We're going to take a look at virus groups 6 and 7 in this lecture. So group 6 viruses are called retroviruses. The reason they got the name retrovirus is they're able to do reverse transcription. So the RE in retro is standing for reverse, and the TR stands for transcription. So that's how they got their name. So the most famous retrovirus is HIV. So retroviruses are very unique in that they can do reverse transcription. So they have an enzyme called reverse transcriptase that will do transcriptions in the reverse direction. So if you remember the central dogma, going from DNA, transcribing it, making our messenger RNA, and then translating the messenger RNA to make our protein, that's what all cells do. But what HIV is able to do is they can do reverse transcription. So they can use RNA, and from RNA they can make DNA. Our cells are not able to do that at all since we do not make the enzyme reverse transcriptase. This is the life cycle for making more group 6 viruses. All group 6 viruses have plus single-stranded RNA as their genetic material. And when they get into the host cell, they need to take that plus single-stranded RNA and from that make double-stranded DNA. So they're taking RNA and from that they're making DNA. So that's reverse transcription, and they're going to use the enzyme reverse transcriptase to do that. So there's two steps that happen. The plus single-stranded RNA will be used to make a complementary minus single-stranded DNA to go with it, and then the minus single-stranded DNA we'll just make a plus strand to go with that, and then we end up with the double-stranded DNA. So both of these steps are done by the enzyme reverse transcriptase. Our cells are not able to do this because we don't have this enzyme, and so the group 6 viruses have to bring the enzyme reverse transcriptase into the cell with them. Once we have the double-stranded viral DNA, that gets inserted into the host cell chromosome where it will stay until the cell dies. So the double-stranded DNA will then be transcribed to make the viral messenger RNA or the plus single-stranded RNA. And remember, the enzyme we use to do transcription is RNA polymerase because we're making RNA. And it's DNA dependent because we depend on DNA for the instructions to make the RNA. And then the host cell ribosomes can translate the viral messenger RNA, make the viral proteins, and then make the capsid. And then the last step is we just need to make more of the plus single-stranded viral RNA. So we'll just take the double-stranded DNA, and then from that we'll make the plus single-stranded RNA. So we're going to use RNA polymerase because we're making RNA, and it's DNA dependent because we're depending on DNA for the instructions to make the RNA. So an example of a group 6 virus is HIV. So HIV eventually will cause AIDS or acquired immunodeficiency syndrome. There are two strains of HIV. There's HIV-1 and HIV-2. Most people that are infected with HIV have HIV type 1, and that's the one that has spread around the world. Interestingly, HIV-1 actually came from a virus in chimpanzees called SIV, which is simian immunodeficiency virus, and we believe that SIV jumped into humans probably somewhere around 1910 in West Africa, and we think it was probably the person was exposed while butchering a chimpanzee for bushmeat for food, and the blood got into a cut, and that's probably how SIV got into a human, and it mutated and became HIV. So this is a picture of what HIV virus looks like. It does have an envelope, and you can see the spike proteins. And then inside the capsid, you can see the reverse transcriptase enzyme that it brings with it. That's there in red. This is a picture of the HIV life cycle. The HIV virus will infect some of our white blood cells. Most importantly, it affects the helper T lymphocytes or helper T cells. So the virus has an envelope with these spike proteins. The spike protein that is used to attach to the receptor on the white blood cell is called GP120. GP just stands for glycoprotein, and just 120 is just talking about how much it weighs. The receptors that HIV virus binds to on our cells, we have the CD4 receptor, which we'll talk about more when we get to chapter 18. And then the other receptor it needs to help enter the cell is the CCR5 protein. Remember, that's the protein that the scientists altered in the twins that we talked about in the CRISPR discussion. So once the virus gets into the cell, it's going to use the reverse transcriptase to take the plus single-stranded RNA to make double-stranded DNA. And then that viral double-stranded DNA gets inserted into the white blood cell chromosome. And that viral DNA is now called a provirus. So remember when we talked about the temperate bacteriophages, that they also insert their DNA into the bacterial chromosome, and that's called a prophage. But these are not phages, they're viruses instead, so that's called a provirus there. And then we can make more virus, and HIV will then leave the host cell by budding and pick up the envelope on the way out. Okay, group 7 viruses, our last group. This would be the hepatitis B virus. And group 6 and group 7 viruses are the only ones that have reverse transcriptase and can do reverse transcription. So hepatitis B virus is also able to do reverse transcription like HIV. So hepatitis B virus has a very unique genome. It uses double-stranded DNA. And we'll take a look at a picture here in a minute and just show you how it didn't quite finish making it. Hepatitis B virus is spread through bodily fluids, most commonly through contact with blood or sexual transmission, and can also be transmitted from mother to child as well during birth. The virus infects the liver and can cause some liver damage, and sometimes people will get jaundiced or icteric, where their mucous membrane, skin, whites of their eye will turn kind of a yellow because the bilirubin builds up in their blood because the liver's not working. Some people end up with a chronic infection or persistent infection with hepatitis B virus. Most people do clear the virus, get rid of it, but some people don't. And if you have these chronic infections with hepatitis B virus, it will cause cirrhosis of the liver or scarring. This is a very ugly looking liver. It should not be all lumpy and bumpy like that. So there's a lot of damage and scar tissue that's been formed in the liver. The other thing that hepatitis B virus can do is it is an oncovirus. So it is possible to cause liver cancer if you have a chronic or persistent infection. Here's the hepatitis B virus. It does have an envelope, so you can see the spike protein sticking out there, and then you can see the capsid, and then inside we've got the double-stranded DNA. Also attached to the double-stranded DNA, we have the enzyme reverse transcriptase that it brings in with it because our cells do not have this enzyme. So the double-stranded DNA of the hepatitis B virus was not finished being made. I have no idea why it's like that, but it just is. So one of the strands here in blue, if you follow that around, it is complete, but there's a nick in it. So it's not joined together there at the end. And then the other strand in purple of the double-stranded DNA is incomplete. There's only about half of it there. So when the virus infects the cell, here's the life cycle for making more group 7 viruses. So here's our genetic material of the virus, our double-stranded DNA that's gapped. So we've got one strand that's complete, but there's a nick in it, and then the other strand is incomplete. So there's only about half of it there. So when the hepatitis B virus gets into the liver cell, your cell will see that double-stranded DNA that has not been finished, and our cells will actually finish copying the rest of that double-stranded DNA and make it complete. So that's really nice of our cells to do that for the virus. So to fix the DNA, we need to make more DNA, so we're going to use a DNA polymerase. And it's DNA dependent because we're depending on the DNA to give us the instructions to complete the double-stranded DNA. And then once we have the double-stranded DNA, we just need to make more viral proteins and more of the viral nucleic acids. So the double-stranded DNA, we can transcribe that, make the plus single-stranded RNA, our viral messenger RNA, and we're going to use a DNA-dependent RNA polymerase to make the viral messenger RNA, which can then be translated by the ribosomes, made into the viral proteins, and that'll make the capsid. And then from the viral messenger RNA, the plus single-stranded RNA, we will then take that and make the double-stranded DNA that's gapped. So we're going from RNA and then making DNA from that. So that's reverse transcription. So that's where the enzyme reverse transcriptase that the virus brought in will work. So that step is completed, and then we'll just put everything together and make more hepatitis B virus. And then you don't need to memorize this picture, but it's just showing you hepatitis B virus infecting a liver cell and going through and producing more virus, which we drew out on the other slide. You don't need to memorize these next slides here with this chart of different viruses, but it's just interesting to look through and see which viruses are more closely related to each other.